Hello everyone, welcome back to my channel. Today I'll be discussing on a very important chapter, that is molecular base of inheritance. Under this chapter, I've taken a very important topic, that is transcription in eukaryotes. Before getting detailedly into what is transcription in eukaryotes, let us know the basics. This is a commonly asked question. How does DNA provide the information required by a cell to function properly? We know that DNA or genes help in synthesizing or instructing for the synthesis of proteins by a cell. You know that our body constitutes lot of enzymes, hormones or antibodies, and these are the chemical messengers which transfer certain signals from one part of the body to the another. I guess you got the answer. Transcription is the first step in the synthesis of proteins from the DNA. Now let us know about the transcription unit. Under transcription unit, you have to understand a very important concept, central dogma of molecular biology. That is, DNA can synthesize RNA with the help of process called transcription. RNA can synthesize protein with the help of process called as translation. Now, my dear friends, what is transcription? Transcription is the copying of genetic information from one DNA strand into RNA by RNA polymerase. This process is called as transcription. A question can arise in your competitive examinations maybe. Unlike replication, only one DNA strand is copied to RNA in transcription. Why? We know that replication is a process of copying DNA strands, or it can be also called as cloning, wherein transcription is a process of synthesizing DNA into RNA. Now let us see into the reasons. We know that DNA is double-stranded. So reason number one, let us assume that both the DNA strands acts as a template strand. How many RNA sequences are formed now? Two different RNA sequences are going to form. So these two different RNA sequences will complicate the transfer of genetic information. Reason number two, these two different RNA sequences produced will be complementary to each other, which is going to form double-stranded RNA. And this double-stranded RNA will not lead to the process called as translation. So from the translation, what we have to obtain? Proteins. Now let us look at the different regions of a transcription unit in a DNA. Now let me explain you about the structural gene. You can see here, isn't it? Since DNA is double-stranded and wherever the structural gene is present, within that region, the DNA strands are having two different names. Since RNA polymerase, now RNA polymerase is an enzyme which comes and binds only the promoter region to start the process called as transcription. The strand with 3' to 5' polarity is called as template strand, and the strand with 5' to 3' polarity is named as coding strand, even if it is not coding for anything. In transcription, you can see the promoter region and terminator region, which is placed on either side of the structural gene. The second region is promoter region, and promoter region is a DNA sequence which is located towards the 5' end of the coding strand, that is towards the upstream. Promoter region is a binding site for the RNA polymerase enzyme to initiate the transcription process. RNA polymerase enzyme catalyzes the polymerization only in one direction, that is from 5' to 3' end. Terminator region, it is a DNA sequence located towards the 3' end downstream of the coding strand. It provides the stop signal and defines the end of the transcription. Before going deep into the process of transcription, let us understand certain terms which is needed to understand the process of transcription. The first one is the gene. The functional unit of inheritance is known to be a gene. Although there are many genes present on the DNA, it is difficult to define a gene in terms of a DNA sequence. Now you can see here the DNA sequence that codes for tRNA, transfer RNA, or rRNA, that is ribosomal RNA, is also a gene. Under gene, we have to learn about the term cistron. Now, what is cistron? Cistron is a segment of DNA that codes for a polypeptide. Polypeptide is a polymer of amino acids. Under cistron, there are two different types of gene. The first one is called as polycistronic gene. The second one is called as monocistronic gene. Poly, by the word it says many, mono means single. Polycistronic gene is found in prokaryotes and bacteria, which is a lower organisms, and it can code for several proteins. Mono means single, as I've already told you, and it is seen in eukaryotes, and it codes only for one single protein. As I've told you, monocistronic genes which only appear in eukaryotic organisms, that is complex organisms these are, and it codes for a single protein. And it consists of two sequences. One is coding sequence which is called as exons, which you can see in red color, and one more is intervening sequence which is called as introns, which you can see in blue color. Also, we have to know the types of RNA. So there are three types of RNA: messenger RNA, which can also be called as mRNA; second, transfer RNA, which can also be called as tRNA; and third one, ribosomal RNA, which can also be called as rRNA. The main function of mRNA is it provides the template to make the protein. The second one is transfer RNA or tRNA. The main function of tRNA is it reads the genetic code and transfers the amino acids for the protein synthesis. And third one is ribosomal RNA. The main function of rRNA is it is involved in the structural and catalytic role in the protein synthesis. Now coming to the process of transcription. The following steps are: first one, initiation. Here, the RNA polymerase, which is an enzyme, only binds to the promoter region. And also it binds to the initiator factor, which is called as sigma factor. And now it can initiate the process, that is transcription. Step 2, elongation. Here, the RNA strand starts getting longer, and the RNA polymerase recognizes the DNA nucleotide and keeps on adding RNA nucleotide to the growing RNA transcript. The RNA polymerase walks on only one DNA strand. The third step, and that's the last step in process of transcription, that is called as termination. In termination, the transcription is coming to an end. This is how, because the RNA polymerase and the sigma factor, which is coming to an end of the terminator region, is going to fall off along with the RNA transcript. Now, the RNA polymerase is going to bind to a new factor, and what is that factor called as? Rho factor or terminator factor, and this stops the transcription process. Coming to the last part of the session, that is complexity in eukaryotic transcription. This information is very important both for your theory as well as for your competitive examination part. Now, why is it called as complexity in eukaryotic transcription? This is because eukaryotic itself is found in the complex organisms, that is higher organisms like humans. Now, there are two important major complexities. The first one is the mRNA, which has been formed after the transcription process. that is named as primary RNA or pre-mRNA. This is because they are non-functional. Why they are non-functional? Because it contains two sequences. One is coding sequence and one more is intervening sequence. I hope you remember, which I've explained, that is exons and introns. Introns are not needed, it has to be removed. We only need exons, and this is why it's called as non-functional transcript, and it is named as pre-mRNA. Now the pre-mRNA has to undergo post-transcriptional modification like splicing, capping and tailing to form a functional mRNA or heterogeneous, processed heterogeneous mRNA. And the second one, the processed RNA, which is functional now, has to be transported out of the nucleus for the next process that's called as translation. The major enzymes involved in the post-transcriptional modifications are RNA polymerase 1, which transcribes the rRNA, RNA polymerase 2, which transcribes a precursor of mRNA, or heterogeneous nuclear RNA, which is also called as mRNA. RNA polymerase 3, which transcribes the tRNA, that is transfer RNA, and small nuclear RNAs, which is also called as snRNA. These three enzymes are very much important in the post-transcriptional modifications in eukaryotes. The post-transcriptional modifications in eukaryotes undergoes the first step, splicing. Now, in pre-mRNA, as I've told you, it is non-functional. It involves both introns and exons. Now, the introns have to be removed, so splicing has to happen. It is a process of removal of the introns and joining the exons together in a specific order. Second step is capping. Once the pre-mRNA becomes functional, that is introns have been removed, to the 5' end an unusual nucleotide is been added, and that is called as capping. It is only, you have to remember, the 5' capping is only added to the mRNA or fully processed heterogeneous RNA. Third step is tailing. Now, tailing is done by adding 200 to 300 adenylate residues to the 3' end of the mRNA. Now, the mRNA is completely protected at the 5' end and 3' end and ready to move out of the nucleus and then start the process translation.